Now that the definitions of soil health are more clearly outlined, we can focus on methods used to increase it. In Module 2, we will discuss many ways to increase soil health. We will cover composting and cover crops, different biological-based soil amendments, biochar for carbon capture, safe use of animal manures, and government assistance programs designed to make these healthy choices more affordable. Our next speaker, Dr. David Johnson, is director of the Institute for Sustainable Agriculture Research at New Mexico State University and adjunct professor at California State University at Chico. He is a molecular biologist currently conducting research in soil microbial community structures. He studies the interactions and interdependence between plants and soil microbes to improve plant carbon capture efficiencies, system water use efficiencies, and reduction of soil carbon respiration. His research is aimed at developing practical and easily adoptable mechanisms to significantly reduce greenhouse gases while promoting sustainable agroecosystems.
This planet is why it is today because of the living organisms that have inhabited it. You can see all through history the influence of different organisms on our planet, and for the majority of the time, it's been microbes. And microbes are the backbone of every organism and ecosystem on this planet. In the human microbiome, we're outnumbered 10 to 1 in the number of cells. We only have about 10 trillion human cells and 90 trillion microbial cells. We have 1 quadrillion resident viruses. We have about 20,000 human genes, but our microbial genes number up to 20 million. So you're less than 1% human as far as gene count. What do they do for us? They do food digestion, they do cell renewal. We used to think they comprise 60% of our immune system. We now think it's 80% or more. They control your appetites and cravings. They do nutrient generation, vitamin synthesis, detoxify carcinogens, prevent allergies, skin diseases, and asthma. And they also turn on and off genes that regulate your brain development, anxiety, depression, autism, arthritis, and emotional behavior. I may have to start over, it locked up. I'm gonna have to reshare. I'm gonna have to back out and start over, I think, here.
It's okay, take your time. I can see it.
There, okay. Okay. Here we go. Oops, that was the wrong one. So as I say, microbes are the backbone of every organism and microbe or living being on this planet. We'll get through this. So what happens when these microbial communities are disrupted through either administration of antibiotics or diet-related issues? Cancer, inflammatory bowel disease, irritable bowel syndrome, surgical infections, liver disease, almost all of these are related to a dysbiosis or a disturbance of our microbiome. We can see since 1950, 200, 300, 400% increase in many of the diseases, asthma, diabetes, multiple sclerosis, and Crohn's disease. We're looking at the diversity of gut microbiota from people from Tanzania and Malawi compared to the US. You can see that the US has much less diversity, and a lot of it's due to the foods that we eat. You can see here, when we remove fiber from the diet of mice generation after generation after generation, we start to lose diversity of that microbiome. Well, you say just bring the fiber back. You see, when you bring it back, you do not restore that diversity. What you have to do is come back with a fecal matter transplant or an inoculation of microbes from a healthy individual and put it into the gut microbiota of the host. And you can see you immediately restore functionality to that system. So if this works in the human microbiome, because we've cured many diseases with this, there's so much research now linking our gut microbiome to the diseases that we have. Kind of simple inoculation of beneficial microbes restore the microbiome structure and function in agricultural soils. Looking at how nature has set this up, you can see that your bare parent material, your soil, your deserts are predominantly bacterial. As you move up this ladder of succession from the deserts up to your old growth forest, you see a change from bacterial dominance to fungal dominance. You also see a change in the net primary productivity, or the amount of biomass that can be grown on a square meter of soil. 90 grams here in a desert to 2,200 grams in old growth forest. Here you can see the influence of fungi. Fungi are critical in this system. This is a real-time photograph of bacteria moving along a film of water on a fungal hyphae. This is where we are right now in our agroecosystems. We're low carbon, bacterial dominant. Where we need to be is right about here, 1-to-1 or greater. This is where your late successional grasses grow best and your row crops. So we need to understand, to stay on this ladder, that there's a certain amount of disturbance that's required in this system. Nature used cows and bison. They would be held in tight herds by predators. They would be moved along the prairies at a regular interval, so they would only take about 40% of the forage and trample the rest into the ground. They would deposit their dung and their urine and their saliva on this system. And as they moved on, the dung would be rolled into small balls by the dung beetles, placed into the soil, and it would be basically composted in place. So what these organisms did, these grazers, these ungulates, as they moved across these grasslands, they were inoculating these systems with biology. Well, can we mimic this in our system on agricultural lands? So we started this out with a project with USDA to do something good with dairy manure. And what we did is we developed a no-turn composting system. This is a Johnson Sioux bioreactor. You use about 70% moisture content in the process. There's no turning involved. It's a static composting process. It's aerobic because you have air columns up through the center of the pile. After the temperature goes below 80 degrees, you add worms, and it takes about 1 year to reach maturity. This one bioreactor will produce about 700 pounds of compost. I know it doesn't sound like a lot, but our application rates now have been shown that we can have efficacy at as low as 2 pounds of compost per acre put as an extract into the furrow at planting. The compost, when it's done, looks like a big chocolate cake, and when you squeeze it, it's a lot like clay, as you can see here in this video. We did some analysis looking at the phyla through the composting process from 160 degrees down to 60 degrees. You can see the ebb and flow of different phyla through this whole process. The firmicutes start real strong, but they end up very small in the final product. Proteobacteria come and go through the system. Bacteroidetes start small, but they end up being a major component at the end. This is looking at 2 compost samples analyzed a decade apart. The top one was basically dairy manure, yard waste, and some wood chips. The bottom one was all leaves. You can see there's a striking similarity in the end product of the compost, even starting with different materials, even done a decade apart. What we see as you go through this process, the diversity increases. This is what it looks like at 4 weeks. This is what it looks like at 22 weeks when most people say it's ready. And this is at 60 weeks. You can see we have a 4 times increase in diversity. Well, we did an experiment with this compost looking to see its influence on plant growth. We did some chili plants and we compared it to 8 of the compost available in our area. What we noticed, we had these 2 on the left are from our compost. One of them had been put into the greenhouse 5 months and watered daily to flow through to see what we could irrigate out or nutrient loss. You can see it was one of the best producers. But when we looked at nitrogen, did it correlate to plant growth? No. We had some of the lowest nitrogen measurements. Same thing with phosphorus, potassium. We thought, okay, it's got to be organic matter. Organic matter did not correlate well to plant growth. What did correlate pretty well was fungal to bacterial ratio. You see about R squared of 0.88. So this put us on a different path, looking at fungi and bacteria. We did another greenhouse trial with chilies, looking to assess carbon and nitrogen partitioning. You see here, we started with a low carbon bacterial dominant system, 0.04 was fungal bacterial ratio, up through all the way to a higher carbon system with 3.68 fungal to bacterial ratio. What we noticed in a low carbon bacterial dominant soil, only 3% of the energy that plant captured went to the plant. The rest of it went into the soil to improve soil carbon content, respiration, or total new nitrogen from the carbon assimilation from that. Tracking each of the, well, let me tell you, the red is the fruit carbon, the green is the shoot carbon, the brown is the root carbon, orange is again that total new nitrogen, the carbon used to form that nitrogen. Respiration carbon was blue, and the new soil carbon is gray. What we noticed as we improved the fungal to bacterial ratio, along with increasing soil carbon content, was an increase in the amount of carbon that would flow to the plant. You can see 21% at a fungal bacterial ratio of 1.6, 30% at 2.3, 37% at 3.02, and 56% at 3.68. So you see it's critical to change that from a bacterial dominant system to a fungal dominant system, but you also need the carbon as you're going along. Here we are in our agricultural system. We're about 11% efficient. This would show us that we have the ability to increase production by 5 times. Does this work in the real world? What we're talking about is a biologically enhanced agricultural management or BEAM approach to agriculture. Looking at the most productive ecosystem, terrestrial ecosystem on the planet is about 2,200 grams of dry biomass per square meter. Cultivated land, for all that we do to it, from fertilizing it, watering it, weeding it, we only net about 600 grams of dry biomass. What we see with BEAM in a transitional system is up to 2,200 grams, and advanced BEAM to 3,200 grams. This is what it looked like when we first started. We spread this at about 400 pound per acre rate. This is what a 1 square foot would look like. It's just a dusting. We're not putting nutrients down. We're putting the microbiome. You can see here a control after 1.5 years of BEAM, planting a winter cover. We had to change from 50 grams a square meter on this crop to 250 grams just after 1 year. Year 7, we're looking at 1,141 grams of dry biomass per square meter. So we've 5 times what we saw after the first year. That's about 387 kilograms of nitrogen. We did not use any fertilizers on these treatments. It's just putting a crop, growing it, and inoculating it at the beginning. 1 time, we didn't inoculate after that. This is a summer cover that we did. It's black oilseed, sunflower, sesbania, forage soybean, and a couple other. You can see here on June 23rd, it's about 4 to 6 inches tall. 29 days later, it's about 5 feet tall. This is 17 days later. We have black oilseed sunflower, about 7.5 feet tall. This is very unusual. Black oilseed sunflower usually only gets about shoulder high. This crop on the right is sesbania. It's a crop that's native to southeastern California, southwestern Arizona. It's a legume. It also associates with mycorrhizal fungi. It's a great cover crop. It's great for shading out any weeds. The first season we grew this, it was 4 feet tall. The second season, it was 6 feet tall. The third season was 12 feet tall. We see a continuous change in the ability of these soils to grow a plant as you bring this biology back. This is another trial we did. You can see this is where I live in the desert. This is on a desert sand. On this particular experiment, what we did is grew cover. We inoculated at the 400 pounds per acre rate. We grew a cover crop, we cut it, we haul it off, and then we plant it again. A total extraction, trying to tax and see just what the biology can do. The first season we grew this cover crop was 800 grams. This is the second season. This is a winter cover crop being grown at 1,608 grams. This is the third season. At 2,190 grams of dry biomass per square meter on this plot. That is as much production as the most productive terrestrial ecosystem on the planet. And this all happens within 10 weeks. You plant this in November. It doesn't really get going until mid-February, and it has a crop like this by the end of April, the first part of May. Looking at each season, the change in the dynamics of growth. The yellow is the uninoculated, the green is the inoculated. You see, we almost had double the productivity from the inoculated. As you go up through here, you'll see at the end, if you inoculate, it's much more productivity than if you don't inoculate. Now, cover crops are you can see here, but to come in with inoculation, you have much better productivity. This was 14.37 tons per acre of dry biomass. Now that's a pretty good yield on a forage crop. You'll also notice from the start to the finish, we saw a 5 times increase in net primary productivity. This is what the soil looked like at the beginning. There's no A horizon. This is what the control looked like. We developed a 3-inch A horizon and a 7-inch A horizon in the compost treated. It makes that much difference to come in and bring this biology into the system. We saw from the compost treated soil, these are all the phyla, doing a metagenomic analysis on this system, looking to see when we add the compost, what are the organisms that really come forward. And you can see there's 21 different phyla that are much higher than either our control or the soil that we started with in the desert. So we need to see that diversity is the currency of survival on this planet. This is looking at metagenomic analysis. We saw about 2,750 species of microbes. About 193 of them came to the top with different characteristics and abilities. What we saw was about 21 different nitrogen fixers, both free living and symbiotic. We saw nitrogen cycling, nitrogen degraders. We saw carbon cycling. Now, carbon cycling is critical. Carbon is the energy resource for this system. And what you need to do, the carbon that you need in the soil needs to be a functional carbon. That's what makes these systems work. We saw metal oxidizers. These are the ones that free the elements up from your soil parent material. You see phosphorus solubilizers. For the most part, most of you have 40 plus years of phosphorus in your soils. It's inaccessible because of the soil physical chemistry. Yet these microbes can free it up and make it available to the plant. We saw microbes that degrade pesticides and antibiotics. We saw microbes that produce phytohormones. We saw some that are very strong influence in quorum sensing. Now, what we notice in this is quorum sensing. Quorum sensing is when a group of microbes get together, either a single species or multi-species microbes, and they work as a community, expressing genes as a community that they can't as a single organism. And we saw a very strong upregulation of quorum sensing genes in the systems where we had restored that biology. In fact, it was up to 10 times higher than the expression level of just your normal housekeeping genes, which is highly unusual. So what are the benefits of this regenerative agriculture? We see you're able to build carbon up quicker than most of the scientists are seeing. West, Nigley, and Law from 0.2 to 0.7 tons of carbon per hectare per year increase. We observed 10.7 tons of carbon increase per hectare per year. That's a 0.25% increase in soil carbon each year. We see the potential to do 0.5%. Now, this is what it would look like on ours. We had a relatively good progression, 0.25% increase. That's 10.7 metric tons of carbon per hectare per year, or 37 metric tons of CO2 per hectare per year. Now, most of you probably know Gabe Brown. The first 13 years, he tried no-till. He tried crop diversity. He tried cover crops. He was only able to increase his soil carbon about 0.1%. Once he got into multi-species cover crops and livestock integration, he reached 3% soil organic matter, and he stopped using nitrogen fertilizers. He saw an increase rate of 0.56, which kind of substantiates what we were seeing at 0.5% increase per year. This is what his soils look like. For the last 3 years, he's had only 5 inches of rain. We visited him this last July, and we drove into his pastures. These grasses were as high or over the height of his hood on his truck, a 4x. And that's only 5 inches of rain. He has completely changed the dynamics of soil and how he can grow forage. We also have to look at these systems, healthy systems, the respiration changes. This is back to that greenhouse test, looking at what we're calling soil carbon respiration, or the percent of the initial carbon respired. There's only about 2 grams of carbon in this particular treatment, but 55% of it was respired. But as you increase that fungal to bacterial ratio, and as you increase soil carbon, only 11% of that carbon is respired. So you have a much more carbon use efficient system as you bring this biology in. So you see a 10 times increase in beginning soil carbon mass, but only a 4 times decrease in relative soil carbon respiration. As you bring this carbon back in the soils, you also see, for the first 1% increase in soil carbon, you quintuple the water storage capacity of that soil. This helps your plants not go through so much stress in your water and dry cycles. And it also stores up water and lets you use it more efficiently. We're seeing historical depletion of micronutrients in our food, from 40 to almost 90% in the vegetables, fruit, meat, cheese, and dairy. This is mostly due to what we've done to our soils and how we outstrip the ability of these soils to provide those micronutrients. What we saw in the first 20 months of this study, bringing BMAC into the system, was an increase in the availability of manganese and iron 1,100%. We also saw magnesium, calcium, zinc, and copper from 40% to 80% increase in availability. We also saw increase in nitrogen, phosphorus, and potassium. Again, we're not putting any fertilizers in on the system, and we've only inoculated the system at the very beginning. And our soils are very high pH. And if you notice, iron and manganese, the two that we saw the biggest increase, they should be decreasing in availability, but due to the soil physical chemistry. But once you bring the microbes back into this system, they can free it up and make it available to the plant. What we saw in our yields, our cotton production was about 2,400 grams of lint per acre in the bean, compared to 1,170 in our conventional in this area. In our chili production, we saw 20.8 tons compared to 10.5 as an average in our system. If you're organic, you take about a 30 to 40% loss, because what you're doing is you're practicing organic processes using natural, conventional processes using natural nutrient amendments. We're trying to move away from the amendment. These soil minerals and elements are there. They just don't have the biology to free them up. And you can see organic could be here if they start to bring the biology back and concentrate on that. This is our, after 1.5 years of bean compared to 200 pounds of nitrogen per acre after 1.5 years. This is July 17th, 27, our cotton crop. August 2nd, August 29th, we had 6-foot cotton, but that doesn't mean you have cotton. unless you do. We had about 5 bale cotton on this pasture and about 3,000 pounds of seed per acre. This is about a $2,000 gross crop an acre. Our chilies in 2018, we had 17 to 21 tons of chili per acre. And the farm hands were waiting for these to be ready because they knew they never had been sprayed or never had been fertilized. This is, we wanted to look and see if we could reduce the amount from 400 pounds down. And we did an analysis looking at applying an extract in a furrow at planting. On the right, we put the extract on top of the soil and watered it in. On the left, we sprayed it into the furrow as we planted the seed. You can see the significant difference in the response. In fact, there was over double the productivity in the injected soil compared to a topical application. What we noticed is there was no difference, though, between 2 pounds, 4 pounds, 10 pounds, or 20 pounds per acre. So a 2-pound rate can make quite a bit of difference. We tried this on corn production in Wilcox, Arizona, looking if we inoculated corn seed with the extract as we planted them. We planted it at 1.52 pounds of compost per acre rate, or about a 20 gallon per acre injection rate. This is what it looked like under the microscope. For every square foot that we applied this, we had about 83.4 million bacteria and 10.4 million fungal spores. This is what we got. We had a beam approach here, biology only, at that 1.62 pounds per acre. This is the beam plus 15% of the conventional nitrogen at about 38 units of nitrogen per acre. And this is a conventional at 256 units. Now we had a sub-treatment on this of a multi-species cover on the left and a single species triticale cover on the right on every treatment. You can see the multi-species had much more influence on the crop that followed compared to the triticale on every treatment, even the treatment that had the 256 pounds of nitrogen per acre. As you can see, as you go across the field, same thing all the way across the field. What we saw in this, our conventional produced about 217 bushels of corn. Our bean plus 15% did 217.8, just a little better than the conventional, reducing 85% of the nitrogen used. And our beam only with no nitrogen produced 203 bushels. We saw only a 6.6% yield loss with just the bean. So the profit was $88 greater on the bean plus 15% and $60 per acre on just the bean. What we also noticed was nitrogen flow. Those plants, or those microbes, had to fix a lot of nitrogen. We saw over 116 pounds of nitrogen per acre fixed by the microbes that we put in the soil in the bean, 84.5 in the bean plus 15%. And in the conventional, we saw a loss to the environment, either to the air or to the water below this pot, of minus 122 pounds of nitrogen. On the mixed species covers, we highly recommend the mixed species. Have more productivity, you can see 477 grams compared to 361. But it's actually cheaper to use the 7-way. You get more biomass for your money, 34 cents per gram of dry biomass per square meter compared to 41. The Haggertys in Western Australia, they're on the front lines of this. What they do is they use about a 2 pound per acre inoculation rate of the compost that they buy. You can see here on the right the influence it has on the rhizosphere. Now, they are producing 3 tons of barley. This is conventional dry land, Western Australia, basically a desert situation. So they're producing about 3 tons of barley per hectare in a region that conventionally gets 1.4 to 1.5 tons. Now their conventional neighbors are going out of business and they're buying their land largely because they're salinizing their soil. But when they come back with the biology, they're able to regenerate and reclaim these soils. This picture on the left, they've already harvested the barley, as you can see. They got a late fall rain, and in nice, neat rows, exactly where they injected this inocula is a C4 grass that they hadn't seen for 4 decades in that area. Many of the government officials said it wasn't native to the area, but that seed bank survived, and once the biology was right, these plants could grow. That's how powerful the right biology is. On your farming, you need to start thinking outside the box. This is Adam Chappelle in Arkansas. He's using 68 row spacing on his cotton. You can see he has a nice cover that he's rolled down and planted into. This is what that cotton looked like. This is corn in Iowa, Lauren Steinlich. He's done 60-inch row cotton, corn, but he's planted a cover crop, an intercropping in between a rye, grass, buckwheat, chickling vetch, oilseed radish, and rape. Pecan farmers, this is how they do it in Las Cruces. They disc it, they spray it with herbicide, they water it, and they start all over again. Or they just let it grow up in weeds. What you can do is put a cover in, something like this. We're seeing many benefits for the pecan growers, where if they do put a cover crop in and they do keep cover in that soil, they reduce the soil temperature from 120 down to 80. You get better nut fill, higher quality nuts. You don't get the wilting in the process. So you need to think biology, you need to think restoring that biology, and these cover crops are the food resource for that biology. We did some research with Peter Vick in Arizona State University, looking at the difference between conventional and AMP grazers, those that do the multi-paddock grazing. And what we found, as you increase fungal biomass in these systems, your standing crop biomass goes way up. Very good correlation. You can see fungal biomass is critical. We also noticed that bacteria are negatively correlated to this. So the more bacteria, the lower the productivity. We also noticed respiration rates, kind of going back to what I showed you before. In conventional systems, the respiration rate was much higher compared to AMP, and AMP was much higher compared to a natural ecosystem. What we noticed, as you reduced the respiration rate, your relative respiration rate, you increase biomass production in the system. So it's a system that we're trying to bring back. It's not just one organism. It's not just the cover crops. It's a whole system that you have to be thinking, bringing the biology back, bringing that flow of carbon into the system to support these organisms. Nancy Ranney in Corona, New Mexico has seen, she's gone from 3 to 4 grass species to 45 grass species in a decade. She reduced her medicine bill by 90%, supplemental feed bills by 66%. She had originally built swales to capture the water for stock tanks, but they stopped filling, but her aquifer started going up. Again, we are restoring a system. Alejandro Carrillo in the Chihuahuan Desert south of us, he started out with this, 250 acres to support one cow. He started using proper grazing management. He's brought that land back now to 20 acres to support one cow. He's brought back grasslands in what used to be grassland, but was desert after we mismanaged it. So that's about all I have. Align yourself with nature, observe what you do, and try something different every year. And what I want to say one more thing about soil health. It's trying to measure it is very difficult right now. It's like you going to a doctor and having him do one test on you and tell you that if you're healthy or not. It takes multiple tests. We are at the very threshold of starting to understand this right now. And there's many experiments you can do or samplings you can take, but they're very expensive right now. We're working on that to try to figure out, is there a much cheaper, better way? But you start bringing the covers in, you bring that biology back into the system, it reacts totally different. Farmers, that 3% soil organic matter is the threshold that this happens at. But having the right biology there is key. And this compost gives you 2,700 different species. It's not just 1 or 2 species.
Our next speaker, Dr. Kristine Nichols, is a research microbiologist with a background in studying sustainable agricultural systems. She is the former chief scientist of the Rodale Institute and is currently conducting research with the MyLand company. Her current research is focused on stimulating growth of native algae cultivated at farm locations for the purpose of using the algae as a soil amendment applied through irrigation water.
And I definitely want to thank all of the previous speakers. As was just mentioned, all of this really integrates well together and is very much integrated. And so when we're looking at foundational types of systems, as Dr. Johnson was just talking about, this whole idea of our microbial communities actually acting as kind of the foundational stimulators and formers of soil and soil health. I've done an extensive amount of work with mycorrhizal fungi as well. And so looking at how all of these organisms are going to be functioning together is incredibly important. One of the things that I'm working on now is this project looking at microalgae that we are adding to the soil system and utilizing it by applying it through irrigation water. Okay, I need to hold on one second. For some reason, it is, I apologize, I've had this problem before, but it's not advancing. There we go. Okay. So basically, what are microalgae? A lot of us may be sort of familiar with algae, mostly on a macroscopic level. And so microalgae are similar types of photosynthetic or photoautotrophic or autotrophic organisms. They are unicellular, but many of the species of microalgae that you have are also mixotrophic, meaning that they can be autotrophic or photoautotrophic as well as heterotrophic. So they can utilize the formation of their own sugars via photosynthesis, and they can also utilize sugars coming from other organisms, including coming from plants. So these are things that we don't have to completely make sure that are applied to the soil surface and are being exposed to sunlight, but we can actually utilize them. One of the projects I'll discuss towards the end is a system in which we're applying through a subterranean drip system. So they're going to be, the microalgae we're applying are going to be acting heterotrophically. The microalgae are microscopic, so they range in size from a few to a few hundreds of microns, but again are extremely small, not like the common algae that you would be familiar with seeing in more aquatic-based systems. And what's interesting about algae, and a lot of people would look at the desert Southwest and wonder, why are we focusing on algae? Aren't they based in aquatic systems, and desert systems are dry systems? How can that possibly be the case? And really, algae, especially microalgae, are found in ecosystems all across the globe, not just in desert systems, but in forests, in fields, in aquatic systems, and even in Arctic systems. So some of the driest environments on the planet are actually Arctic systems, because it is so cold that you don't have water crystallizes, as opposed to, so it's very dry, as opposed to having water that is in the liquid form. So they're very dry systems in places like Antarctica, and you will have microalgae that will be growing in these systems. So microalgae have always been important in desert ecosystems as well. They help in the formation of biocrusts in these desert ecosystems. There are hundreds of thousands of different species of microalgae, and they are found in concentrations of about 3 to 100 million cells per gram of topsoil, or about a teaspoon of topsoil. The common species that are utilized are listed here, primarily chlorella and Haematococcus, and Spirulina are the ones that are found most frequently and are used more on an industrial basis. So microalgae have been found in a number of types of agricultural systems. They've been used in rice-based systems for hundreds to thousands of years. They are currently used in row crop systems, have been found and applied in wheat, corn, sunflowers, peas, chickpeas, cotton, sugar beets, sugarcane, and produce crops, primarily lettuces, cabbage, peppers, tomatoes, and radishes, as well as ornamental flowers and trees. So they can be utilized in a number of systems because they are found universally in a number of systems. They are frequently applied as either living or dead cells, most commonly as dead cells, or as extracts of the intercellular and extracellular biomolecules. And these can be applied to seeds themselves. They can be applied to foliar tissue or to soil with or without other products. So you can utilize some of the other things that have been talked about today, including mycorrhizal fungi and some of the technology that Dr. Johnston was talking about, where you can be mixing some of these other microorganisms with the microalgae. What's important about the microalgae is that they contain a high concentration of protein in their cells. They also contain a high concentration of lipids and glycerols, and a high concentration of various types of vitamins and antioxidants and minerals. So they are almost like, in some forms, a concentrated fertilizer pellet containing many of the nutrients that the plants, as well as the biological communities, microbial communities, are going to need to be able to grow. And so when you look at the proteins and the lipids, all of these molecules are carbon-based molecules. So they all can also help to contribute to the organic matter that is in the soil. Microalgae can be produced in bioreactors or living soil factories, where they can be utilized as a biofuel source. They're also utilized in pharmaceutical and cosmetic industry. They can be utilized as food or feed. And so there are many types of uses that microalgae can have in the system. If we look at the history of microalgae, as I mentioned, prior to the 1900s, microalgae have been used in rice production for hundreds to thousands of years. But they haven't been really explored individually until Muriel Bristol did some work in 1919, and then in the 1950s, mass culturing techniques, as you can see here, culturing in various types of containers, bioreactors. This is utilizing some carboys, some plastic containers to be able to do the culturing, but you can utilize glass tubes or plastic tubes to be able to do the culturing. But this mass culturing really came into place in the 1950s. There was more commercialization of mass culturing in the 1980s. And then research has really been flourishing since about 2008. And so really focusing on the functional roles of microalgae, what some of the benefits are to the system. MyLand is a company in South Phoenix that is basically has designed a unique production system, bioreactor system, in order to be able to culture the microalgae into high density concentrations that then can be applied to the field through irrigation systems. And we've been really working on this technology since about 2019 and 2020. So there's been some big breakthroughs that are very recent in this technology. If you look at the benefits of microalgae when looking at what may be happening with the plants, they've been found to increase root and shoot length, dry weight, yield, germination rates, cellular respiration, floral production, photosynthesis, chlorophyll production, and resilience to various types of stressors. Also, you have various types of microalgae, the cyanobacteria in particular, that are able to fix nitrogen. And this nitrogen fixation can be in a very high amount within the system. So it can be a way of helping to supplement or even replace synthetic nitrogen application by utilizing this biological source. It's also interesting to find that these biomolecules that are produced by the microalgae have some very strong roles in helping to control various types of pests and diseases, including bacterial pests, viral pests, nematodes, fungal pests, and protozoas. They're also very important, as has been discussed several times, the role of soil aggregates in the system. And these microalgae can produce these compounds that are actually important, like the glomalin that was mentioned earlier, that can help to put soil particles together, bind them together into soil aggregates. And when you have better aggregation in the soil, you have more porosity, which means that you have greater water infiltration rates and better water retention capacity in the system. Microalgae can also help to break down various types of compounds. They're utilized a lot in various types of bioreactors, are utilized in wastewater systems in order to be able to clean the wastewater systems. And as I mentioned, they're high in nutrient content, so they can provide a large amount of nutrients. Some of these biomolecules are also able to solubilize phosphorus to help in phosphorus availability, and also to help in chelation of various nutrients like iron and calcium. So they play multiple different roles with the different types of biomolecules that they are able to produce, and how that's going to be able to help with the system and moving forward. Now, why are we focusing on microalgae? Well, one is looking, as I discussed, what all of these benefits are to the system and what they can provide. In addition to the microalgae themselves, when we're utilizing the system that MyLand has designed recently in the last few years, this system is actually a way of being able to apply live microalgae. As I mentioned, microalgae has been applied in most cases as dead microalgae or dead cells, or as the extract of these biomolecules that can play these important roles and have the benefits that I just mentioned. But with the MyLand system, we're utilizing live microalgae. And the benefit of this is that when the algae are applied to the soil, they will continue to replicate in the soil, both growing autotrophically or photoautotrophically, utilizing photosynthesis, or heterotrophically, where they can utilize sugars that come from other microorganisms or coming from the plants. And they will continue to replicate in the soil and keep growing into fairly large concentrations in the soil. In addition, those biomolecules that I discussed continue to be excreted, so they can continue to play those roles while they're replicating and growing in the soil on an almost continuous basis. And the way that the MyLand system functions is that we are actually culturing indigenous microalgae. So we are looking at for native microalgae that are in your soils. So they are adapted to that environment. But then it will also be where they are applied as living cells and can continue with these roles and functions that you're going to be able to have happen. And the way that the system functions is that we can continue to apply microalgae to your fields during each of the irrigation events that you might have, so that you're getting a continuous supply and building up the microalgae in the soil into very large numbers. In addition, in the way that they act as living organisms, they also have roles to play as dead organisms. So once the microalgae die, they can act as a nutrient and food source for other organisms. So their dead bodies may be consumed by other microorganisms, by bacteria primarily. And then as those bacteria are breaking down the microalgal cells, they are going to be releasing the nutrients, especially nitrogen and phosphorus, and even potassium that may be held within the bodies of those microorganisms. So when we're looking at these benefits, it isn't just what's going to be happening with the living cells themselves, but also what may be happening with the dead cells. Again, as they're growing in the system, those cells are dying. They are also helping to create microporosity or porosity pockets in the soil, which that porosity is going to help with aeration and with water movement. And they are going to stimulate the secretion of various types of compounds when they are consumed by other microorganisms, especially the bacteria. So the bacteria will use them as their food source to create various types of compounds that are going to be beneficial to the plants and also help to generate organic matter. We've talked a lot today about the value of organic matter to the system. So the microalgae act as organic matter themselves because their cells contain carbon. So both the living and the dead cells can be organic matter, but they also produce biomolecules or exudates that they secrete that are also part of the organic matter components. And then when the dead cells, those carbon compounds that are part of the bodies of these organisms are consumed by other organisms, they are also producing organic matter fractions. So it's helping this entire microbial community, as Dr. Johnson was talking about, and helping to be able to build up organic matter in the system. Now, what we have here is some of the ways in which we can culture the microalgae in the laboratory system. But when we're talking about the benefits of utilizing live indigenous microalgae, and indigenous just means native to the system, so, or native to your farmscape. So what we're doing is we're actually going out and taking soil samples and water samples from the farms that we're working on and farmers that we work with, and culturing the microalgae in the laboratory, identifying the species or the individual species or the multiple species that may be beneficial, most beneficial to be culturing in our bioreactor. So we're going to utilize, again, these live indigenous microalgae that are able to replicate and form symbiotic relationships with the indigenous microbiome because they are more adapted to that environment that they came from. So they're more adapted to the soil chemistry and physical structure and mineralogy, but they are also adapted to the various types of microorganisms that may be in that soil environment. And so then when we reculture them in a bioreactor and reapply them, they're going to be able to respond more quickly to various types of stressors that may exist in the system much more quickly. Again, they're also, the organisms themselves, the microalgae themselves, as both living and dead cells, are carbon sources, and they're going to be composting quickly because they're going to be consumed by other organisms to build up organic matter in the soil. Again, they produce various types of polysaccharides to help to form soil aggregates, which is going to help in irrigated systems where we can potentially reduce the amount of irrigation water that's needed, and also to help reduce the amount of evaporation that may be happening in the desert southwest. And this was a very important role that microalgae played natively in desert systems when they were part of these biocrusts, where they would help with water management. They would reduce evaporation in desert systems, and they would also help with infiltration into these desert systems. In addition, as I mentioned, they secrete various types of nutrients. They also can secrete some hormones, antioxidants, and other molecules that can help to spur plant productivity. They can also help to produce carbonic acid, which has been mentioned as far as some of the roles that it can help in some of these desert systems to help to break down carbonate crust that you may have and loosen soil and generate better porosity. So we've got, again, multiple roles that they can play. And in particular, when you are applying these living indigenous microalgae, they are very adapted to the system and can play these roles very quickly. And because they're living, they're going to continue to play these roles while they're living, and even providing benefits when you have the dead cells. This is one of the systems that MyLand has designed. This is our Series 30 system, which was designed in 2019. And basically with this system, essentially what you have is a shipping container that goes out into the field. And then within that shipping container, we have a number of these bioreactors. And so the algae are cultured in these bioreactors with artificial light. And you can see in the bottom picture here in the lower picture, basically as we're refilling the tanks with water, the microalgae that have been left behind in the tanks can be a source of providing inoculum in those refilled tanks. So it is a way in which we're basically bubbling the microalgae back in so that they can start growing. So it's a sort of a continuous loop generation type of a system. And then these tanks will be emptied and injected into the irrigation water. At this particular site that is pictured here, this farmer actually uses above ground sprinklers as part of the system, as well as flood irrigation in some areas. And so we're able to incorporate the microalgae into both flood irrigation systems as well as above ground sprinkler systems. So again, it's microalgae where we're not running into the issues that sometimes have been of concerns in the past when utilizing microorganisms in irrigation systems, where they are going to be clogging the irrigation lines. This is something that we have found that they're unicellular growth, and as long as we keep a system going where we add CO2 and bubble the system, we don't get large colony cluster formation that could potentially clog up the irrigation system. So it's a really great way, and that's what MyLand has done. Is basically we're not marketing the algae, but we're marketing the system. So the Series 30 is one of the systems that we have. The 1100 series is another system that you have. It's for some smaller scale operations or smaller scale fields, rather than the system that is designed within the cargo, the shipping container. And it is basically designed to where you may have no electricity, or you may have less access to water or other things like that. So in more remote locations, we can utilize this 1100 series system. It works in a very similar way where the tank is acting as a bioreactor. We put media that is nutrient media that can help to culture the algae in that system. And then we can either utilize a gravity type of injection system or a pump type of an injection system in order to be able to get the algae into the irrigation. So in this case, we're doing a flood irrigation from canals. So being able to insert the algae into that system. One of the things that the company has invested in is actually doing some research activities. So we have a large scale field experiment that is going on at the U of A Research Center in Maricopa, so the University of Arizona Research Center in Maricopa. We have this study where we are able to test essentially 3 different concentrations of algae, as well as a positive control, which is the nutrient media that is used for culturing the algae, but no algal cells added. And then a negative control, which is just water by itself. And we are able to test 4 crops. We have 2 cropping seasons. So our fall or winter cropping season, we had broccoli, head lettuce, celery, and romaine lettuce. And in our spring cropping season, we had watermelon, cantaloupe, bell pepper, and processing tomatoes. And the plots were laid out in a randomized complete block design, and each of the, as you can see, each of the lines, each of the columns were treated with a particular type of algae, but there were 4 blocks in each of the columns for the different types of crops that we would have in there. We did this in order to be able to, again, test what might be happening with the different concentrations of microalgae on a number of different crops. What we would see, so as you can see, the picture up in the right-hand corner is the microalgae that we isolated from this farm and cultured in our bioreactor system. And then we would apply this microalgae. This is our broccoli crop. And you can see differences in the plant growth and productivity with the utilization of the microalgae. So the 1x is sort of our standard concentration, 2x is 2 times the standard concentration, and 5x obviously is 5 times the standard concentration. And then again, as I said, we've got our negative control, which is water only, and our positive control, which does contain the nutrients that have been utilized to culture the algae. And those nutrients, we add them to the irrigation system. This is a subterranean drip irrigation system. So again, it's something where a different system than the flooding system, the sprinkler system that I mentioned earlier. So this is very flexible as far as our ability to utilize a number of different types of irrigation systems. And the media nutrient solution that's added is only added in the concentrations that is needed to grow the amount of microalgae that's being added. So we're not adding a huge amount of micronutrients and macronutrients, but we are making sure that any of those background nutrients are controlled for by our positive control. So this was for broccoli. This is for romaine. And what we saw, which was kind of unique with our romaine crop, was that we also saw a greater density and size, diameter, especially with the taproot here, with our romaine lettuce roots in the treatments that were treated in the algae treatments as opposed to our controls. So we had a noticeably longer and denser root system in that 2x treatment than we would see in our control treatments. So again, we're seeing some benefits of being able to utilize that in the system. I don't have data for our summer crops, the watermelon, cantaloupe, cabbage, or the watermelon, cantaloupe, tomatoes, and peppers yet. We just have finished up our harvesting for that. One of the things that I would like to invite you to do is we do have a book called Smart Cultures by Dr. Mark Edwards, who's on faculty with the University of Arizona. And we have various copies of this book that MyLand would be happy to send you if you want to email me. So my contact information here is k.nichols@myland.ag. And if you want to email me, I will make sure that you get sent a copy of this book. The website address here is our web address for MyLand. Again, that web address is MyLand, MYLAND.ag. With that, I want to thank you.